proteins in these bilayers and gives you sufficient signal to noise over these multi-layers to actually generate the diffraction patterns that can be used to detect where proteins are in these multi-layers. So this is a way to, again, look at proteins associated with the bilayer through diffraction, where you need the increased signal and the ordering in the 2D lattice. Okay. So what we're doing, so now I just want to get into some examples of how we make our measurements and what we like to do with them. So when we're designing protocols for the tethered bilayers and protein reconstitution, at this point it's really an empirical exercise. So we want to be able to go rapidly through designing experiments where you can test different phospholipids, different additions of cholesterol, other kinds of lipids, ligands, but you want to be able to test the system with various different conditions. You want to be able to do this and at the same time monitor the membrane bilayer integrity, and you want to be able to get some idea of the surface concentration of the components, and particularly the membrane protein components. And then ultimately you want to be able to detect the proteins you want bound to your surface for small molecules, and then determine some kind of affinity for those surfaces. So and again, ideally to monitor it close to real time. So there's a number of methods one can use to interrogate surfaces, and the two I was going to highlight today are surface plasmon resonance, which can give you some information about surface binding and affinity, and electrical impedance spectroscopy, which again gives you some idea of the membrane integrity because it measures ion mobility across the membrane. There are other possible other kinds of measurements like AFM or TURF. TURF's a fluorescent measurement where you can look at sort of tracking particles like lipids that can give you some idea of the fluidity of the membrane or the movement of the protein, but